Hi, I'm Sheila Mahoney from LifeSciHub. Very happy to have Jeff Davidson of Alpha Kappa Statistics here with us today. Jeff is a longtime biostatistician in life sciences, has a tremendous body of work, having worked in various capacities for sponsors such as Hoffman LaRoche, Shire. He worked at Accenture for a bit, and also Endo Pharmaceuticals, among others. And in the last 15 years, most of the levels at which he has worked has been VP of Biometrics or Global Head of Biometrics. And Jeff is now an independent consultant. Welcome, Jeff. Thank you for joining us.
Thank you, Sheila. I really appreciate you giving me an opportunity to share my experience, and hopefully we can answer all your questions today.
And you've had some extremely interesting experiences in your career, I think, starting from the early days and then into the later 2000s. Can you share some of your experiences that you would like folks to know about?
The FDA at this time changed the rule to you must show that when a placebo is given to the subjects, that you can differentiate your product from the placebo. So only placebo-controlled trials would be accepted for the pivotal trials for an application. This was identified at the time when the submission had occurred for the product, and of course the pharmaceutical company was very, very concerned about how to move forward. They had done all of these studies using the old methodology, and now they had to use new methodology. So there was a quandary. How do we conduct these trials? If we give a placebo to these subjects, they're going to drop out. We're not going to have what we need in terms of the numbers to be able to compare. I had experience in oncology where one looks at survival curves. You see, for example, when subjects enter an oncology study, you have basically 100% survival. As you go through the trial, depending upon whether you are on the active product or on some comparator or best practice, best treatment available, because it's unethical to not give subjects anything when they're in an oncology trial, you want to look at the survival curves. And if you can show that your product has a higher rate of survival, then you have demonstrated that your product is better than what is currently available. So it took this methodology and transferred it to the pain area and worked with the clinicians to come up with a design that has now become the standard. It is called the randomized withdrawal design. And what we did at that time was we said, well, if we're going to use placebo, we need to make sure that the subjects come into the trial with their pain under control. So they call that part of the trial the open label titration to effect phase. So everyone is given the experimental treatment and they allow enough time for the pain scores to demonstrate that the subjects are responding or feeling relief from the pain. At that point, a randomization occurs. Subjects are then treated in a blinded fashion with either the active drug, that is the experimental drug, or placebo. And for those subjects who need to have rescue medication, that is permitted. It's typically a very low level pain medication. Otherwise, it would confound the ability to demonstrate that the active drug is working better than the placebo. In this design, the subjects go through the trial, and the placebo subjects generally don't see pain relief. You don't want them to see pain relief. You want to show that your drug is working and that without a pain control, that there is no response, but you're still giving them that rescue med. You hope that they'll live through the trial and that they won't disappear on you and drop out. But in many cases, there were dropouts. this was something that the FDA understood was going to happen, and they accepted this. So the randomized withdrawal design, the withdrawal part of it, is withdrawing that medication at the time of randomization, and then watching as the two groups demonstrate a level of pain relief that can be scored at the very end to compare with what their scores were at baseline. So if you imagine, there's a survival curve that appears from these data where your active group, for the most part, stays in, and the placebo group, many of them will go through to a certain point, but some of them will drop out. So you'll see those curves really separate. And the comparison was made at baseline and at the endpoint, so that you would compare those two response rates. And this design is now the standard for how pain studies are conducted, and I was very thrilled to be part of the seminal discussions and coming up with the design for how that would be studied. That was an exciting part of my career, and that occurred in the early 2000s.
Yeah, what's interesting about that is that the creativity that you employed in that case, that sure, statistics is pretty much about math and probabilities and very clear equations and data, and how much creativity can there possibly be? And yet, that's a great example of utilizing creativity in that capacity in order to solve a fairly serious problem. Would you agree?
And I'm glad you mentioned that creativity is something that when you think of the work of a statistician, you generally aren't thinking of someone who's creative. You're thinking of someone who understands the theory, who understands how statistics are used in clinical trials, but the creativity part of it is really where the most enjoyment comes. When you can think about a particular problem, a particular question that comes from the researchers, and then you provide that insight so that they then know how to go forward and design the trial. And it's a great collaboration. And the collaboration is what is most needed in order for a clinical program to be successful. When you have statisticians and clinicians working together, you're going to have the best opportunity for success. You have to do your best to be accommodating to the advisory committee members, and you have to have a great team. And when all of that works together like a well-oiled machine, you can be successful. And the best occurrence during an AdCom is that they don't dwell on that question, that they move past it. So we were able to move past that question. When AdComs go south, it's when a question is asked and a sponsor member responds, or one of the experts responds, and it devolves into an hour-long discussion about something that may not be meaningful, or on the other hand, it may be meaningful. You have the information if you can produce it, and if you present it, and the people on the advisory committee are able to digest it and accept it, then once again the question will go away and you move on to other questions. And that's the ideal situation in an advisory committee meeting. So that was another example of an advisory committee meeting that I participated in. So they are exciting. They're full of tension. They're full of angst until they're over, because you don't know what questions are going to come. But if you're prepared, if you've gone through the gauntlet and had all of the tough questions asked of you in the preparation phase, you can answer any AdCom question that comes your way.
Yeah, wasn't it Dwight D. Eisenhower who said, plans are useless, but planning is indispensable? Or something along those lines.
Those are words to live by.
Jeff, thank you for all of that great information. You have incredible experiences and a extremely robust body of work that is extremely valuable to many drug sponsors. I'm just wondering if, as you look out at the current trends of biostatistics and some things that sponsors need to be aware of, things that are happening either with FDA or other regulatory agencies, can you speak a little bit about what you see the trends and the hot topics are and how best to be prepared for that?
Great question. Thank you for that. I would say one of the trends is that the FDA is looking more and more at what we call a tipping point analysis. And that is when a sponsor has prepared and has presented the FDA with the information on their pivotal studies, typically 2 adequate and well-controlled studies that are going to be the basis of the approval, the FDA will come back now more often than not and request a tipping point analysis. So what is a tipping point analysis? The tipping point analysis is used to determine how robust your results are. And if you think about a fulcrum, if you look at any situation where you have a balance, the weights are equally distributed on both sides of the balance scale.
Mm-hmm.
At what point can you remove the information or the response? How much of the response can you take away before you've tipped the results so that they are no longer significant? That's the simplest way I can put a tipping point analysis. And there are mathematical methods that are used to perform a tipping point analysis. So I think in today's FDA environment, the tipping point analysis is very important to determine whether or not you have a result that is close to being non-significant or one that is so robust that the tipping point would require removing a lot of the response in order for non-significance to occur. That's one of the areas that I see being critically important for sponsors to be looking at, for statisticians to be aware of, and for preparations for the review.
That's really a great explanation. Thank you so much. And it definitely highlights one of the remarkable things about you as a biostatistician and what you have to offer. You have that unique combination of incredible technical abilities, experience, as well as the ability to communicate it in a way that makes sense to lay people. Because you just helped me in less than 30 seconds understand what a tipping point analysis was with a perfectly apt metaphor. And I think that communication, scientific communication, is a skill set all its own. And I think in biostat, that is pretty rare.
If you don't have the communication skills to enable others to understand what you're saying and to get your point across effectively, and communication, more importantly, involves listening. So one of the things that you have to learn if you're going to be a great communicator is be a great listener. And I pride myself on listening more than I do talking, because you can learn a lot from that. So thank you for noticing that. And I know that under circumstances where a sponsor needs that type of communication with the agency or within their own company in order to help people inside understand what the impact of statistics is in their particular research efforts, that's where I'm able to make an impact. The scenario is one of great intimidation. That's the only way I can put it. You will have lined up across the table a team of FDA reviewers, and there's typically, well, they try to match you in numbers. So if you bring 12 people, they'll bring 12 people.
Oh.
But they are the authority, and they're there to protect the public, and they have an important job to do, and that can be very intimidating. They're going to ask very difficult questions. You have to be knowledgeable. You have to know your program. You have to know the studies. You have to know the data, and you have to be able to answer in a very objective way in order to give the FDA the information that they need. I've been fortunate enough to work on roughly 30 products that have received FDA approval during my career. Many statisticians go through an entire career and don't work on a single approved product. So I've really, truly been blessed with having had that experience and in multiple therapeutic areas. And it's priceless. So what I'm going to do is support sponsors who need that help, because at this point, I could walk away into the sunset, but I really feel that there are lots of products that I can help move towards being marketed, and that's where I will spend my time now. Medical affairs teams need to have a statistician, just like the clinical development teams. When a product is being marketed, when it's in the peri-approval phase and they're looking at getting publications out there, a statistician can be extremely helpful. There are lots of ways in which the data can be exploited and explored, and there are many, many opportunities to prepare manuscripts, abstracts, and posters. I have had the privilege of, in one of my careers, in one of my opportunities, I worked to build a medical affairs team from scratch, because the company, large pharmaceutical company, at that time didn't have a medical affairs biometrics team. And in that effort, that team was able to produce clinical trials, actually, that were used by the medical affairs team to prepare publications, and also to exploit the data, as I said, to look at what's in the data, to see where publications can be prepared. And once again, you're using a lot of your imagination, you're looking at the data, you're massaging, you're determining how that data can best be used to present the information in such a way that it makes sense, that it stands up to scrutiny, and that it can help the public understand and the medical community understand what this product is all about. And medical affairs teams are very good at doing that, and I'm very good at helping them to accomplish that.
Fantastic. Well, I think we're just about out of time. Jeff, I wanted to thank you so much for participating today and telling us a bit more about Alpha Kappa statistics.
Thank you very much for inviting me, and it's been a pleasure to sit and talk with you about something that I've enjoyed doing for 30-some years and hope to continue to do, and to help LifeSciHub as well.
Wonderful. Great. Thank you very much.